This is Audiommunity, a podcast about our body's never-ending fight with the outside world. Today, we're talking about immunology in plants. I know even less about bananas than I do about plant innate immunity. I'm speaking with Pamela Ronald, professor of plant pathology at the University of California at Davis, about her work in plants' innate ability to recognize pathogens and the use of genetic engineering to artificially instill that resistance into bananas, a staple crop for millions of people around the world.
In East Africa, 100 million people rely on bananas as their staple food source. And there's been a devastating disease that's sweeping through East Africa.
And we talk about the controversy surrounding the use of genetic engineering to produce food crops.
I'm not a proponent of genetic engineering. I'm a proponent of sustainable agriculture. And I think many of the discussions about genetic engineering are really a distraction from sustainable agriculture.
The audio of the phone call isn't perfect, but I hope you'll stick around, and I hope you'll enjoy this episode as much as I do. Welcome to Audiommunity, Episode 6. I'm Kevin Bonham, and today we're doing our first guest interview, and I'm joined by Pam Ronald. Pamela Ronald is a professor in the Department of Plant Pathology and the Genome Center, and the director of the Laboratory for Crop Genetics Innovation and Scientific Literacy at University of California, Davis. And she also serves as the director of grass genetics at the Joint Bioenergy Institute. Pam is also the author of Tomorrow's Table, Organic Farming, Genetics, and the Future of Food, and she contributes to all sorts of online publications about plant biology, plant genetics, genetically modified organisms, and the like. And the reason that I asked Pam on today is because she is also has a long history of investigating plant immunology, and that's something that nobody that's normally on the podcast knows anything about, despite the fact that my research for my PhD was pretty closely related, at least from the animal side. So welcome, Pam. I guess I should have said that at the beginning, so we can get your voice recorded. So I guess to start, just like to talk about your background. How'd you get started in science? What was your introduction to it? And
thank you, Kevin. It's a great idea to have this podcast, and it's a pleasure to talk with you. And let's see, how did I get started in science? I am a California kid and grew up in the summers in the Sierra Nevada mountains. And so we did a lot of hiking and backpacking and botanizing. And so I've always been very interested in biology. And also I really liked math when I was a kid. So that probably drew me in there.
And so you did your undergraduate at
Reed College.
Oh, right.
Yeah.
Yes, absolutely. Okay. So you did your undergraduate at Reed College, and that's up in Oregon. It's actually a place that I would love to end up if I could get a job there at some point. But so you studied biology at Reed?
I did.
Is it biology major, or is biochemistry, or just straight up biology?
Just biology. It's a really small college, so we have the very basic majors.
Mm-hmm. And then you did a master's in Sweden, is that right?
Yeah, when I was at Reed, I was very interested in plant microbe interactions, and actually Mount St. Helens blew up while I was there, and I was able to work with some scientists that were studying recolonization of Mount St. Helens by mycorrhizal fungi, which are really important microbes that form symbiotic relationships with plants. And then that got me very interested in that field of work. And so then I went to Sweden to continue looking at plant microbe interactions.
Okay, and sorry, you can tell that I'm new at this whole interviewing thing.
Oh, you're doing better than I would. So that's fine.
Okay, so what did you study at Uppsala University?
I studied the interaction of mycorrhizal fungi with plant hosts, and the idea back then was, why are plants susceptible to some microbes, but then they recognize other microbes and then they defend themselves against those microbes? And of course, some microbes, you don't want to launch a defense response because you need to have a symbiotic relationship. So I did some really basic studies trying to understand that, using pine roots, which is not the easiest system to work on. And so when I came back to the United States to work on my PhD, I was very intrigued with some of the work of Brian Staskowitz, who was looking at that time, peppers and tomatoes and their interaction with pathogens. And so I was very interested in trying to understand what were the genes in the plant that were critical for recognizing the pathogen, and what were the genes in the bacterial pathogen that trigger the response in plants. And so at that time, what we've been eating, breeders had been breeding for resistance genes for over 100 years. So virtually everything we eat has these genes for resistance that allow the plant to recognize and respond to the microbe. And it's economically and environmentally efficient because we don't have to spray any types of insecticides. And so I was, of course, also an environmentalist like most biologists. So I was very intrigued with this. And at the time, no one knew what these genes were, so we'd been using them for 100 years. And so it was a lot of excitement in the field trying to identify those genes for resistance.
Right, and you mentioned you want to try to understand why some microbes are recognized as pathogens and some are either not responded to or are recognized as friendly. Do we know, are there any receptors that are actually recognized, for instance, the symbiotic species, do they recognize the microbes as helpful as opposed to pathogens, or are they just sort of passive?
Yeah, it's a really fascinating story over these last 20, 30 years. So there's a very important type of interaction, nitrogen fixation, and certain plants can form symbiotic relationships with certain bacteria, and the bacteria is able to trigger the formation of these very important structures in the root. And we now know that the plant requires receptors that are very similar to the resistance genes. And so there's a lot of parallels between plant pathogenesis and symbiosis.
Cool. All right. So you did your graduate work at UC Berkeley, so went back to California, but then you did your postdoctoral work at Cornell. Is that right?
Yeah.
And you're still sticking with the sort of host pathogen, host microbe interactions. It seems to be a theme that runs through most of your career.
Yeah, we do a few other things as well. We work on tolerance of environmental stress, and also interested in looking at cell wall biosynthesis and modification. But those are the three main projects.
Cool. So in 1995, the part that's sort of most interesting to me as an innate immunologist looking at the mammalian system is in 1995 you published work on this receptor we're going to talk later in the paper about, this XA21 gene that's a pattern recognition receptor in plants. And we've had an episode now on pattern recognition receptors in mammals, the toll-like receptors, and some others as well. But I just wanted to sort of ask you, because like I said, I know absolutely nothing about this field from the plant perspective, as I'm sure is the case with most mammalian immunologists. So how are innate immunity in plants and mammals the same, and how are they different? Are there major areas of overlap? We know the receptors are sort of related, but conceptually, do you draw parallels in terms of how they're doing the recognition? What are the sort of ligands they're recognizing and how they respond?
Yeah, they're really very, very similar. And at the time, in the early 1990s, in animals also, these receptors hadn't been identified. So it was really an interesting convergence of two fields coming together. And we now know that the plant and animal receptors are really very similar. The plant receptors have typically a leucine-rich repeat domain, a transmembrane domain, and an intracellular kinase domain that's fused into a single receptor kinase. And as you are well aware, many of the animal innate immune receptors have the leucine-rich repeat domain that is attached to the kinase domain through different modules. So they're actually separate proteins.
Right.
So that's sort of one structural difference. But the LRRs are very similar, very large leucine-rich repeat type domains. And we know that many of the ligands that they recognize are similar as well. So, for example, TLR5 recognizes flagellin.
Right.
And FLS2, that was isolated in about, I think, the year 2000 maybe, also recognizes flagellin.
Oh, cool.
So even the same molecules are identical.
Right. So, and is that an example of convergent evolution? So, leucine-rich repeat domain is probably a structure that's very old in terms of evolution. But do you think the recognition of flagellin is it convergent evolution, where both plants and animals figured out, hey, flagellin's a really great target for an innate immune receptor, or are they actually genetically related going that far back?
Well, I've heard it argued both ways, actually, that because the leucine-rich repeat domains are really very nice protein binding domains and modular, that it was a sort of convergent evolution event that they were just utilized by plants and animals for this. But you can also imagine it the other way, that a billion years ago, these things perhaps started in some primordial form and then over time just continued to diverge so that the animals now have separated the receptor kinase modules, or perhaps the plants have fused the modules together.
Right. And it's actually, I know there's also the hagfish have a sort of version of adaptive immunity that uses modular leucine-rich repeats as a way of generating what mammals use Ig receptors for. And I think we're probably going to have an episode about hagfish adaptive immunity at some point, because that's super cool. And another thing I know nothing about.
That sounds good.
Cool. So you discovered this XA21 in 1995 after you'd started your own lab, and you've sort of been working on it ever since, it seems. So you already mentioned the sort of structure and the intracellular domain fused to kinase domain, but it sounded like from this paper that we're going to read, I'm maybe jumping ahead a little bit, but you still don't know what the ligand for this is. You know it's coded for by a particular bacterial operon in this bacteria, Xanthomonas. But so do you have ideas about what the actual ligand is, or did I misread that entirely?
We thought we had the ligand, and we published a paper on it, and last year we retracted it.
Oh, that was the paper that you... Okay, right.
Yeah, and so we're back. We were, of course, very discouraged and back to square one, but we've made some good progress, actually, and we have a candidate that we're really quite excited about. We do know that there are genes that are critical for activation of XA21-mediated immunity in the bacteria, and for example, we know that there's a sulfyltransferase that's critical. And so our model has been for a long time now that sulfyltransferase actually modifies perhaps a small peptide, tyrosine sulfation, and then that is secreted outside the cell and triggers the immune response. And we're still very attached to that model, although, of course, very aware that we could be making mistakes, but it's pretty interesting because sulfation is really important for mediating many receptor ligand interactions. So, for example, this CCR5 co-receptor in humans is critical for recognition of HIV, and CCR5 has to be sulfated in order.
I just lost your video feed. All right, so sorry, you were saying CCR5 needs to be sulfated in order to be recognized, or in order to be bound by HIV.
Yeah, and then humans that have a deletion CCR5 are resistant to infection by HIV. So it's a very, very interesting story. So we're just in general very interested in this model, and so we're looking at that.
Cool. And I just want to point out the whole retraction story, because for people that don't know the story, that might be kind of a shocker, but you have a really great post up on our blog, the Food Matters blog. We'll put a link to that in the show notes so people can go read the story of that. But it's sort of retraction is sort of something every scientist fears, and it's a really scary thing to confront, but it seems like the way that your lab handled it is sort of the best you can hope for, and is an example of scientists being good scientists as opposed to what we normally associate retractions with. So
well, thanks. It was definitely not easy. It was very difficult for all of us, and no one likes to make mistakes, but it was really important to us to clear the record, and we also want to keep building on the work, and so we're moving on. So we're excited again about the possibilities.
Cool. All right. So so you mentioned some of your other lab's interests. So I'm aware of the sort of flood tolerance stuff in rice that you've been doing. So you want to just say a little bit about that? You don't have to get into the weeds on it, but just sort of.
Sure. So I started working on rice after I finished my PhD, because I wanted to work on something that was important to the world and also would intrigue me for a long, long time. And rice feeds half the world's people. And so I thought, well, if you can make just small changes in productivity of rice, you can have positive effects on many, many people. And so that's why I started working on rice. And in about 1995, I was really fortunate. One of my colleagues brought me into a very exciting project. So we had just isolated XA21 using the map-based cloning approach, which at that time was quite unusual, especially in rice, which is quite a large genome. And so we thought we could try to isolate another gene that he had been mapping for many years using this approach. So he had been working on a submergence tolerance gene. And this is a really interesting trait because most rice varieties will die if they're completely submerged underwater for more than 3 days. And it's a problem because 70 million people live in flood-prone areas and are losing their crops to flooding every year. And this trait was discovered many, many years ago, probably about 30, 40 years ago in an old Indian variety, but it hadn't been used because the variety doesn't produce well. And so my colleague David McKill, who was here at UC Davis, we actually met at Cornell. He was aware of breeders that had been trying to introduce this trait doing conventional breeding, and they never succeeded in developing a new variety that farmers liked. And the reason is that when you do conventional breeding, you're introducing a lot of genes at the same time. It's not a precise approach. And so conventional breeding brings in a lot of undesirable traits. It's very imprecise. So we thought, okay, let's try to isolate the gene. Then we could introduce it either through genetic engineering or marker-assisted breeding. So we set up a map-based cloning approach in my lab for that gene. Dave had already isolated several markers. So we were able to isolate a large region, and we made a physical contig over the region. And my postdoc started sequencing it, and we found a very interesting gene that was an ethylene response transcription factor. And he was able to show that it was rapidly upregulated after submergence. So we thought that was a very good candidate. So we have a very good rice transformation system in the lab. So we introduced that into a variety that normally is drowning in water. And it was really remarkable. The plants were able to tolerate complete submergence for 2 weeks.
Wow.
So that indicated that we had indeed isolated the gene, which we named sub1A. And then my colleague David McKill, at that time, several years earlier, he had moved back to the International Rice Research Institute. So he was able to take the genetic information and carry out a marker-assisted breeding approach with his colleague. And this is an approach where you actually use positive selection and negative selection. So you just select for the gene of interest, and then you select against genes from the older variety. And they carried several years of field tests, and they were able to develop varieties that farmers have really liked quite a bit. They yield 3 to 5-fold more under flooding. And last year, 4 million farmers grew this submergence tolerant rice.
So that's really cool, first of all. But so just because you sort of touched on introducing these genes and we're talking about selective breeding and some genetic engineering topics, I do want to sort of touch briefly on some of the sort of GE controversies. And it's not really the focus of this podcast, but I just wanted to sort of talk about a little bit about genetic engineering as a strategy for research as opposed to a strategy for actually making things that people are going to eat, because obviously there's a lot of political controversy around that. And I've written a lot on the topic, and I think it's ridiculous that there's all the controversy around it. But I just wanted to talk. So you said that you introduced this gene into cultivars of rice that were better for farmers, right? So can you explain a little bit about how you get a gene that you want into a different variety, and how that might differ from previous breeding techniques, and how is that similar or different than some of the genetic engineering is in the news?
Well, there's many different types of breeding techniques. These days, as the public starts to learn a little bit about genetics, they think of conventional breeding and they think of genetic engineering. But it's actually, of course, more complicated than that. Conventional breeding includes virtually all different types of genetic techniques right now. And you can argue it started 10,000 years ago with primitive domestication, which is where our ancestors just collected seed and replanting it. So that itself alters the genetic makeup of agricultural ecosystem once you're saving the seed and if you're saving seed from lines that have some favorable characteristic. And then, of course, over the last 300 years, plant genetics has accelerated quite a bit with the discovery of hybridization and the discovery of Mendel's basic laws of genetics. So now virtually everything we eat is developed through some method of genetic alteration. And that's because what humans are doing is they're developing plant varieties that grow in artificial systems, which are farms, and they need to be high yielding. You want them to be tolerant of drought. You want them to be resistant to disease. You want them to hold the fruit onto the plant long enough that you can come pick it. So that's why geneticists have been in business for so many years. I think the only thing we eat that's probably not genetically altered in some way is here on the West Coast would be Pacific salmon, for example. On the East Coast, you probably have some wild mushrooms. We have wild mushrooms out here too, maybe blueberries, but there's really very, very few things that have not been genetically altered in some manner. And some of the older varieties of genetic alteration include hybridization has been around since about the 1940s, which takes I don't want to get too long-winded, but there's hybridization. It's been around the 1920s. You take two very different varieties and you make crosses. There's things that you can do embryo rescue, so you can take two different species of rice, for example, and cross-pollinate them. But normally that hybrid seed would die because they're incompatible. But scientists then can baby them along through tissue culture and then create new combinations of genes that didn't exist before. Two different genomes. Grafting is two different genomes, two different species. There's random mutagenesis where you take seeds and you can dip them in a carcinogenic solution such as EMS, or you can irradiate them and create random mutations. And then you come up with 30, 40, 50 mutations, and then the breeder will sort of sort through them and pick out lines that they like. And so that's very, very random. So what marker-assisted breeding is more precise because you can make crosses, cross-pollinate two very different varieties, and then you can track through a genetic fingerprint which genes that you're introducing and maybe get rid of other genes that you don't want to introduce. And then genetic engineering is the most precise, where you basically can take a single gene. And what's become controversial about that is the idea you can take a gene from any species. A bacterial gene, you can put it into a plant, for example. Even though plants often have bacterial genes that have been incorporated over evolutionary time, you can put a wheat gene into rice. So you can do a lot more types of things.
Right. So all of those techniques that you talked about before, the mutation breeding, the crossing, and all that is not under the umbrella of genetic engineering, despite the fact that it's obviously tinkering with the genome. But so it's this one specific thing where you insert a single gene at a time using, I don't know, maybe we should explain this a little bit. So you're using a normal plant pathogen bacterium that has the ability to inject DNA. Is that right?
Yeah. So it occurs in nature, and it's a pathogen that has figured out a method to insert its own DNA into the plant. So looking outside my office, there's a big tree with a big agrobacterium tumor on it. So what geneticists have done is they've taken out the tumor-causing genes and they put in beneficial genes instead. So that's been a very exciting approach, and it's been around for many, many years now. About the first plant probably was developed 30 years ago. And it is, the National Academy of Sciences have looked at this, and it's actually has fewer unintended consequences than many conventional methods of breeding. So, for example, mutagenesis introduces more changes than genetic engineering. Right. So there's no scientific reason for ruling out genetic engineering. It seems to be kind of a gut feeling that people have.
Yes, I completely agree. And there's boatloads.
And you've written a lot about this in a really wonderful, wonderful way.
Thank you.
So I enjoy reading.
Thank you. I'll put links to a bunch of different things from different places in the show notes in case anybody wants to go take a look. But just staying on this topic for a little bit, I just wanted to sort of ask, we'll get to the paper in just a sec, where you have something that's obviously a really potentially wonderful use of genetic engineering. People may have heard about in the news things like golden rice, where there's only a humanitarian purpose, it seems, and people are still a little bit worried about it. But I wanted to sort of ask, there are a few promises that a lot of proponents of genetic engineering often bring up, things like the golden rice example, your example of disease resistance. But I'm wondering, there's some people have talked about things like drought tolerance and a lot of other beneficial things for plants, things that could raise yields, things that could make it much easier to establish food security. And I'm wondering what you think is sort of why some of those provinces haven't been delivered on yet, or if they're coming soon, and also why you think there's such a sort of public resistance to the idea of genetically engineered crops.
I'm not a proponent of genetic engineering. I'm a proponent of sustainable agriculture. And I think many of the discussions about genetic engineering are really a distraction from sustainable agriculture. We really need to use the most appropriate technology to solve a particular problem. And there are real problems with agriculture. We uses a lot of land, a lot of water, produces a lot of carbon emissions. We are producing a lot of food in places we don't necessarily need it, and very little food in places that we really need it. So we really need to foster food security for the poor and malnourished. We need to foster soil fertility, which is a finite resource to have fertile soil. We need to reduce carbon emissions. We need to make agriculture more efficient. It uses less water. And we need to reduce the amount of toxic compounds in the environment, such as reduce the toxicity of many of the insecticides, herbicides that are sprayed on the crops. So these are really the critical issues. And so how we get there, to me as a scientist, is really the technique that we use is less important than whether we are reaching the goals. So, for example, there may be an appropriate, marker-assisted breeding, as my colleagues did for developing submergence-tolerant rice. It was clearly the most appropriate technology. We had the gene. It was a rice gene. You can introduce it through marker-assisted breeding. It can be distributed freely to growers. It got out there very quickly. It's very efficient. There's really no other technology or farming practice that has allowed farmers to grow their plants in these flooding conditions. And floods are expected to increase in frequency and duration as the climate changes. So that is an appropriate technology. And then you think about papaya ring spot virus. So it's a very serious disease in Hawaii. It devastates all the papayas there. And there's no farming method. There's no organic method to control the disease. There's nothing you can spray to control the disease. And so the development of genetically engineered papaya has been really a boon for growers there. And this is essentially you're doing sort of immunization approach. I mean, it's mechanistically different, but conceptually the same, where you're immunizing the papaya by introducing a snippet of a viral bit of the viral genome. And it's worked beautifully. It's been almost 20 years now. It still provides robust resistance. There's still no other technology or farming approach that can control the disease. And so that's what I think about when I think of appropriate technology. You use what's cheap, efficient, environmentally safe, and you move forward with that. And so I think it's really a huge distraction to be concerned about the method of genetics that's used, because as you and I know, the method of genetic engineering is considered no more risky than other methods of conventional breeding. And there's been no incidents of harm to human health or the environment in over 20 years of use. So I think it's really too bad that there has been such a distracted focus on that, because it moves our focus away from really what matters most, is how to enhance the sustainability of our farms.
I'll cut out. That's really great. So speaking of, you mentioned the ring spot virus as a sort of example of plant breeding to resist pathogens. And I think that's a pretty great segue into the paper that you've selected for us to talk about, which is one that I guess came out of your lab or is a collaboration with your lab.
Yeah.
And it's called Transgenic Expression of the Rice XA21 Pattern Recognition Receptor in Banana Confers Resistance to Xanthomonas campestris. And there's some other little Latin name there that I'm not going to try to pronounce. And that's published in the Plant Biotechnology Journal 2014, and we'll have a link to that in the show notes as well. So I guess a lot of what this paper is about is pretty clearly in the title. We already mentioned XA21 is this pattern recognition receptor that you discovered in rice 20 years ago. But why don't you tell us about Xanthomonas and bananas? Why does this matter? Why do we care about bananas, other than the fact that they're delicious?
So banana is a very important staple food crop. It's one of the top 4 or 5 staple food crops. Rice, wheat, maize, banana. Sometimes people will put cassava up there. It's a really important food. We don't think about that so much here in the United States, but in East Africa, 100 million people rely on bananas as their staple food source. And there's been a devastating disease that's sweeping through East Africa. It's called banana Xanthomonas wilt. And I became very interested in this because it's the same genus as the pathogen that infects rice that I've worked on for many years. And we did some analysis, and we found that at least some of the genes that activated the immune response in rice, so these are bacterial genes that activate the rice immune response, are conserved in the banana pathogen. So now that we can sequence the rice pathogen, we sequence the banana pathogen. Our lab didn't sequence it, but other groups sequenced it. You can do comparative analysis and make hypotheses that, well, perhaps the rice gene would function in banana. And so banana does not carry this particular XA21 pattern recognition receptor. So my colleague Lena Tripathi and her husband Jai Tripathi introduced the gene into banana using genetic engineering. And of course, you can't do a conventional breeding approach because these are two different species. And so they introduced the gene into banana, and they carried out inoculation analyses in their greenhouse, and they found that they had 6 banana plants that they showed were completely resistant to the pathogen. So it's really very exciting, and it's preliminary in the sense that these are just greenhouse tests. So we really need to see some kind of field test before we can be sure this approach will work. But nevertheless, I think it's a really good example, and we're going to see more and more of this, that you can take genes from different species and try to solve very important agricultural problems. And banana is very difficult to breed. It's a huge genome. There's very few breeding programs going on, and there are really no robust resistance found in banana varieties now. So you can't even do that kind of work. So this is another approach to developing resistance. And it's nice for the farmer too, because they don't have to change their practices. And it's very important, especially in less developed countries. You can't introduce a whole new farming technique. I mean, perhaps you could, but not very easily. So the farmers can use their same management approaches they've been using for a long time, just with a different banana. So that's the idea there.
Cool. So yeah, we have a running joke on the podcast. In the very first episode, somebody mentioned the Cavendish. I forget exactly even how it came up, but so we've sort of like mentioned bananas in almost every episode. So it's really wonderful that we get to have an episode that sort of focuses on it. So I know even less about bananas than I do about plant innate immunity, but is the banana that's a staple in East Africa, is that the same banana that we eat in the United States, or is it a different?
Cavendish is a banana that widely grown, 47% of global banana production. But I think Cavendish is mostly in South America, and it's genetically identical, whereas in East Africa, they don't grow Cavendish. They're growing many different varieties. People call them backyard bananas and other types of bananas. So the variety that they engineered is not Cavendish. So one of the problems with bananas, especially Cavendish, is since it is so hard to breed, it was developed maybe 100 years ago, longer than that ago, long time ago, and people just love it. So there's essentially one clone that everybody grows, so that monoculture makes it genetically vulnerable.
Right. And that's so the things that are being grown in Africa are not these monocultures. They're actually sort of more genetically diverse.
Yeah. So in Africa, it's very different than these large farms in other parts of the world. In Africa, they're like very small, maybe 1 hectare plots. People are growing different types of bananas, but they're all susceptible to this Xanthomonas bacterial wilt.
All right, so I wanted to ask a few things that came up when I was reading this paper, just because I have you on here. You mentioned in the introduction that there's a number of different things that sort of normal techniques that people can use to guard against this pathogen, Xanthomonas, and some of them make sense. Like you cut off the infected parts of the plant, you remove infected plants and sort of quarantine them and keep them away from healthy plants. But one thing that was in the introduction is that you can remove the male buds of the plants, and I was just wondering if you know why that is protective. What is it about the male buds of the plant are they just particular targets of the bug, or?
Yeah, they're particular targets. I don't even know why, but the disease seems to appear first in the bud, and I don't really know why that would be.
Okay.
I have to ask Lena about that. Okay. She's my banana information source.
Gotcha. One other thing, and this is mostly just for me, but you mentioned here race-specific receptors like the NBS LLR class recognize specific races of the pathogen. I'm wondering, so usually I hear about strains of a pathogen, or I hear about, different species, but is there something about this definition race is that different for plant pathogens, or is that just basically means strain?
It's basically a strain. So for example, we have Xanthomonas oryzae infects Oryza sativa, which is rice. And then there is a subspecies, and then there's often a strain. And so one interesting aspect of XA21 years ago, which is why I started working on it, is that it conferred resistance to virtually all strains that have been identified. And this is very different. And this is way back before we knew that there were these two classes of receptors. Most resistance genes people were working on were race specific. So they would be resistant to one race or strain that's kind of used interchangeably, and they'd be susceptible to another race or strain. And so there was a lot more variability. And so pattern recognition receptors are thought to recognize something highly conserved in all the strains. And there is a lot of hope, or I should say, one hypothesis is that these pattern recognition receptors may end up being more durable because they recognize many different strains and they recognize something conserved and highly important for the viability of the pathogen. But we also now know that pathogens can overcome this broad spectrum resistance by targeting different points in the immune pathway. So I think it's still a little unclear whether this is going to hold up over the long term or not. But right now there are many groups working with the idea that transferring the pattern recognition receptors may be a more durable and stable approach than transferring race-specific receptors.
Cool. All right. Well, that's all I had in terms of my notes. But I was wondering, is there anything that you'd like to tell people about where they can find your work, or is there any other notes that you want to provide, any sort of background that we didn't get to that you think is important?
Thank you. Thank you very much for your interest, and it's really fantastic. You're putting together this website and podcast. It's really going to be so useful to people in the community, and I know how much time it takes, so I think it's really fantastic. And you just received your PhD, is that right?
Yeah, actually I'm walking tomorrow, so I defended last month. And yeah.
Well, congratulations, Dr. Bob.
Thank you. Cool. So so people can find you obviously at Food Matters, which is a blog at Scientific American that we both write for. But where else should people go to find out about your writing or your research? What's the best place that people can?
I can send you the link. The link to my webpage is cropgeneticsinnovation.org.
Okay, cool. Yeah, and we'll put that in the show notes for sure. And all right, excellent. So Pam, thank you so much for joining me, and hopefully this will go up probably in the next week or two. But
okay, well, have fun tomorrow.
Thank you very much. Thanks again.
And thanks.
Have a good one.
Bye.
This has been Audiommunity from emunity.org. We are now on iTunes, and if you like what we do, please go over there and give us a rating. If you don't like what we do, we'd prefer you didn't rate us, but we would love to hear your comments and things we can improve on. So please head on over to the website and leave some comments there. Pamela Ronald can be found at cropgeneticsinnovation.org, as well as all over the web. I'm Kevin Bonham. You can find me on Google Plus and Twitter. Audiomunity has a Facebook page at facebook.com/audiomunity, and you can find all of these links and lots more at emmunity.org. We're supported in part by Harvard Medical School, and the music at the beginning and the end of the episode was composed by Rachel Reinick. Hope you enjoyed it, and we'll see you next time.